Good. Okay. Good morning, everyone. Good afternoon, of course, for those who are on the East Coast, and then good night, probably for those who are in Europe and Asia. So, welcome to our e-seminar series on translational biomedical engineering. I'm very excited and happy to host Dr. Heather Sheardown, the Canada researcher in uh, uh, ophthalmic biomaterials and drug delivery from McMaster University. Um, Before I'll introduce our speaker and then we start the talk today, I'd like to go over a few housekeeping notes. During the presentation, if you have any questions, please write your questions in the Q&A box. It's easier for us to track your questions and ask them from our speaker. Also, there's a poll, as always, available for you to share your thoughts and feedback with us. You can always, of course, email me and Human, and then tell us about your thoughts in private, of course. And then also our next speaker is our very good and very own Tamer Mohammed. Dr. Mohammed is the president and CEO of Aspect Biosystems, a Canadian-based 3D bioprinting technology, who will join us next week. We are looking forward to hearing from him next week. I'm sure it will be a very exciting and interesting talk. But if you want to know more about these seminar series, please follow us on Twitter. You can see our Twitter handle down there, TransLBME. Last but not least, I would like to thank our sponsor, Montreal Transmed Tech Institute, who has been with us since almost the beginning of this e-seminar series. Thank you, Montreal Transmed Tech, for supporting us and supporting this e-seminar series. So with that, I would like to invite Professor Heather Sheardown, who is a professor in the Department of Chemical Engineering with a cross appointment to the Department of Pathology and Molecular Medicine, and an adjunct appointment, has an adjunct appointment with the School of Optometry at the University of Waterloo. I would like to invite Heather to give a talk. But before we let you start, Heather, I have to go over a long list of achievements and bio sketch. So it may take a while, but bear with me. So Heather holds a Tier 1 Canada researcher in ophthalmic biomaterials and drug delivery, and has published more than 140 peer-reviewed papers on this subject. She is currently the Scientific Director of C20, an Ontario research ORF-funded incubator aimed at the commercialization of ophthalmic biotechnologies. Dr. Sheardown was previously the scientific director of the 2020 NSERC Ophthalmic Materials Research Network, which brought together 12 researchers with more than 10 companies aimed at the early stage development of novel material-based treatments for ophthalmic conditions. Dr. Sheardown is the Associate Director of Biomedical Engineering and Advanced Manufacturing, a McMaster partnership with the Fraunhofer IZI, which partners with companies for incubation and commercialization of cell-based therapies, diagnostics, and biomaterials. She holds 18 patents or provisional patents, and is currently the Chief Scientific Officer of the 2020 OptimEyes, a McMaster-based spin-out focused on developing and commercializing a micelle-based technology developed in her laboratory. She runs a large and vibrant research group with more than 10 postdoctoral fellows and graduate students. A pretty translational research, Heather. Very nice And I'm looking forward. And first of all, thank you very much for accepting our invitation. I know how busy you are with all these positions and running such a big lab. But it will be very interesting to hear your thoughts on the translational biomaterials for ophthalmic applications. So I'd like to invite you to share your presentation, your slides, and then the visual floor is yours.
All right, we'll make sure everything is up and everyone can hear me.
We can hear
Can
you
you see
well.
the presentation?
It is. We can see your presentation. Slides are up, and
Excellent.
Then we're all good to go.
It's nice when things work. All right. Thank you so much. This is such a great idea to put together a series on translating biomedical technologies. You know, if this had happened 5 years ago, I'm sure I would have learned a lot rather than sharing all of the mistakes that we've made. So I'm going to talk to you today about specifically 3 ophthalmic technologies and talk about some of the lessons we've learned and the mistakes that we've made. And we've done everything to date through non-dilutive funding, which means grant funding, which means that we're kind of at the end of our road. So we're now moving on to the next phase, which is trying to generate money from outside of government sources. So just because I'm assuming that most of the audience doesn't know a whole lot about the eye, I thought I would just give a little ophthalmic lesson. So the eye, we're all familiar with it, we see, but actually the eye is a really complex organ. It's composed of 2 segments, the anterior segment and the posterior segment. So even within the specialty of ophthalmology, there's anterior segment specialists and even cornea specialists, and then posterior segment specialists, and even that is subdivided. So it's a pretty complex tissue. So the front of the eye, at the very front of the eye, we have the cornea, which is that transparent window that the light passes through. And one of the things that most people don't realize about the cornea is that most of the focusing power in the eye comes from the cornea as well. So if you think about it as a microscope, you've got your fine focus and you've got your coarse focus. The cornea actually acts as the coarse focus, and then it's the lens behind the cornea that does that final focusing on the back of the eye, which is the retina. So because it's such a complicated system, you might think there's lots of things that can go wrong, and in fact there are. And I'm going to talk about systems that we've developed for treating conditions of the front of the eye, so the cornea, and then conditions that we've developed for treating conditions at the back of the eye, which tend to be much more devastating for patients, but also much harder to treat. So this is my standard grant funding kind of slide, or what I use to generate money from the government. You always have to justify yourself. And what I'm trying to say here is that justifying yourself for government funding is very different from justifying yourself for funding for a company or for translation. So when we think about the eye, and I've used this slide for the last 15 years, so nothing has really changed, which is a little bit frightening. But eye disease is increasingly becoming a burden on society.
Heather.
Yeah.
Heather,
Yes.
I'm sorry. We hear echo. I think you have Chrome. This open on Chrome as well. That's probably why we are hearing echo.
Okay. Let me just end the presentation, and I will check Chrome and close it.
Sure, please. And then, you can also unshare your screen.
Do we have an echo now?
We do. We're still.
Okay, Chrome is now closed, so it's not that.
It's much better.
So people say that it's much better. Okay, let's go.
Okay.
I think.
All right. So
sorry.
That's okay. All right. So as I say, my grant funding slide. So eye disease is an increasing burden. It really is a disease of the aging population. And as we all know, the population is aging. So as people get older, conditions of the eye become more prevalent. And we've had, an even greater incidence of ocular conditions through the pandemic as a result of people staring at screens and that kind of thing. So it's multifactorial right now. And what happens when you've got an eye condition is it leads to secondary health problems. So people who are blind or who have vision issues tend to suffer from depression more often, tend to fall more often. The elderly who are visually impaired are admitted to nursing homes much earlier. And when you do the studies, somewhat surprisingly, but kind of tragically, elderly who struggle with vision issues die somewhere between 5 and 7 years earlier than the visually competent population. So this is, like I say, a really great lead in when you're trying to generate government funding, because that's often a lot about, creating a case. So there've been a huge number of new treatments that have arisen as a result of an understanding of the biological basis for some of these diseases. But the problem with the ocular tissue is it's really designed to keep things out. So getting things into the eye becomes a problem, and we need to come up with new and dynamic and inventive ways of delivering drugs to the eye. So what my lab has really focused on is, how do we do this? So how did I get to where I am? Back in 2008, 2009, I was a researcher who had done a little bit of work with companies, was really interested in how things translated, but didn't really understand it. And at that time, I put together the NSERC Ophthalmic Materials Research Network. So for those who are not or were not familiar with these, they no longer exist, unfortunately. These were great networks that brought together academic researchers with company partners to do things that might have translation potential. They were really academic still, because most of the money, if not all of the money in some cases, came from the government, and academic people are very comfortable with doing academic research. So, things happened, but things didn't really move forward. And at the end of the period of the 2020 network in 2014, 2015, we had somewhere more than 30 technologies that we thought had legs, that had potential to move forward and to be able to be used in patients. So my goal then became not to write more papers, because you get really good and comfortable at writing papers as an academic, but things just don't tend to move out of the lab. So at the end of 2020, we decided we were going to put together an application to the CSER program, which was one of the Centers of Excellence programs that was really focused on translation and commercialization. And it was a great experience putting that together. I worked with a number of different people, a number of different companies, had some great advisors, but ultimately the application wasn't successful, which is unfortunate, because it would have really paved the way for us to do some of the things that we wanted to do. And the reason that it wasn't successful in the end was that we had never done it. So I then decided, being, I guess, a little stubborn, that, well, I was just going to do it. So, in that same period of time, I applied for funding from the Ontario Research Fund, more for the research side of things. That one wasn't successful. It came back and they were looking for more commercialization aspects. And then also in 2015, we had an opportunity come up where we could generate some funding from the Ontario Centers of Excellence. And that involved creating a company. So we didn't create a company deliberately. It was more of a creation out of need in order to get funding. But all of this led to us getting a successful Ontario Research Fund grant for the C2020 Innovation Hub. And this grant is really focused on commercialization. All of our milestones are around translation. And we thought, we've got this. We can, within the 5 years of this funding, we've got $4 million $4 million is a whole lot of money. We can most definitely get things out the door. And I'm going to tell you why we didn't and what happened with that. So one of the things I learned really early working with some of the advisors is that it's not about that first slide where it's all about patients and making people's lives better. It's about the numbers. Companies are worried about the bottom line. I have an interesting colleague who worked at one of the large contact lens companies, and he told me he went to his boss with an idea, and his boss said, what are the returns on this going to be? And he said, somewhere in the $1 billion range. And his boss basically said, we don't look at anything that's less than $20 billion. So all of this doesn't include contact lenses, and it was a contact lens idea, but that just gives you an idea about how focused on the bottom line most of these companies are. So the first lesson that we learned very early is that you want to think about what is the potential market? What is the market share? How much of the market can you gain? And what kinds of things can you do to help gain that market? So if you look at these numbers, you'll see that the biggest part of the market share is in antivascular endothelial growth factor drugs. So those, I'm going to talk about those a little bit later, but those are used for treating conditions of the back of the eye. So it seems normal that might be an area that you might go to try and generate some funding and to generate some new technologies. So like I said, at the end of the 2020 network, we had a whole bunch of technologies that we thought we had, potential to move forward. We thought, oh, we're really close. We need to do a little bit more work, but we don't need to do a ton more work. So we had some contact lens technologies. We had some back of the eye technologies. We had some front of the eye technologies. And I'm specifically going to talk about 3 of these here and highlight some of the things that we learned along the way working with these technologies. So, like I said, we started looking at back of the eye disease, and the reason for that obviously had to do with the market share. It's much easier to sell to companies when you think you can garner a big part of the market share. Posterior eye diseases are much more tragic than anterior eye diseases. They tend to lead to blindness. The patients, they're just they have a much bigger impact on the patient. So as a result, the patients are much more willing to put out their time and energy in order to treat them. So the big posterior eye diseases are age-related macular degeneration, which is essentially deterioration of the macula of the back of the eye. Diabetic retinopathy, which is rampant right now because of the increased incidence of diabetes. And then a secondary effect of this is diabetic macular edema. So it's basically swelling of the macula. And then just your normal infections in the back of the eye, which despite the eye being pretty self-contained, can occur and can be incredibly devastating to the patient. And right now, there are some drugs that are used to treat some of these things, but the status quo is essentially a needle directly into the back of the eye. So when I was a child, we used to joke about, if you were really bad, stick a needle in my eye. Well, it's now occurring. It's occurring on a regular basis. And it's painful for the patient. It's inconvenient for the patient. It's particularly inconvenient in the Canadian landscape because you think in Toronto, it's probably pretty easy to find an ophthalmologist. In Montreal, it's probably pretty easy to find an ophthalmologist. But in northern regions, in Ontario and Quebec and in other areas of the country, it's just not easy to get access to somebody who will put that needle in your eye once a month. So, there's lots of downsides to this treatment. It works well in most patients. It requires significant monitoring because if the drug is either cleared or the drug becomes unavailable, the disease can essentially proliferate very quickly. So there's lots of downsides to this. So it really, the ophthalmologists don't like to think of this as a long-term solution, although realistically, we've now been doing this for more than 10 years. So, it would be really nice for something to change. And if you look at the number of injections globally, again, it's growing significantly. So something that we need to be thinking about. So what we did was we looked at kind of the acceptability of injections. Physicians are mostly okay with those. But what if that patient in Northern Ontario, Northern Quebec, remote Saskatchewan only had to go to the ophthalmologist once every 6 months or once a year? Much less bonus them. So could we make something that we would inject into the back of the eye, because we've seen that that's acceptable, but that would last and deliver the drug at a much more a much more regular basis, would give you a much smoother release kinetics, but would allow the patient to go for a much longer time between injections. So these aren't in chronological order. These are in more mistake order. But one of the really cool ones, I think, from an academic perspective anyway, is I had a very talented postdoc who came to me with this idea about combining a very hydrophilic polymer with vitamin E, which has anti-ROS properties. So it mops up reactive oxygen species, which are associated with these diseases, and creating this copolymer that we could inject into the back of the eye and deliver the drug. So he was pretty talented from a chemistry perspective, despite the fact that his background was engineering. And he came up with a really neat way of combining these two components. And then when we inject these into the back of the eye, because we've got this hydrophobic and this hydrophilic component, they rearrange and form not a transparent gel, but a translucent gel that ultimately degrades in the back of the eye, but after a fairly long period of time, and releases the drug relatively slowly because of this oil phase. So the drug release kinetics we used, we actually partnered with Bayer on this one, and we looked at a couple of their experimental drugs. So companies will never give you anything that's real. They'll only give you the stuff that either didn't really work or is kind of experimental. But the release kinetics to me were blow me away positive. So we had, after a 2-month period, we only had 15% of the drugs released with this, and the release curves were very linear. So we were very happy with the potential for this. We tried dexamethasone too. I'm just not showing that graph, and very similar. We actually had even less of that one released over a 2-month period. So kind of taking this out, we thought we might be able to get out to 6 months, maybe not with a protein drug, and that was the caveat there. So we had just started at that time looking at in vivo safety of all of this stuff. So we did safety both in the eye, and then we injected it under the skin just to see what would happen when we injected these things. And by and large, there was nothing histologically that would suggest that these vitamin E PEG systems had any adverse effects in the ocular environment at all. So we put together a paper, and at the same time as putting together that paper, I filed a patent disclosure with my research office, my industrial liaison office, and through a series of missteps, the postdoc was leaving. He was going off to a new job. He wanted to get the paper done and out, and we just didn't follow up. And so the patent didn't happen. So as a result of that, we've got this really great paper, and we've done some work on, the gels since then. But realistically, this is never going to be commercializable because it's not patented. So we've got lovely results, but no company is going to move forward with it because they're not going to be protected in terms of being able to deliver any drug with it. So that was a huge misstep, and it was just a series of events where we did the initial steps and we just didn't follow up and make sure that the final steps happened. So completely my fault. I should have known better, but kind of a lesson learned. And there still remains some issues with these things. So we've looked at, can we change the chemistry a little bit in order to allow us to get that patent that we need? We've actually had significant interest in these materials because people really like the idea of having the vitamin E there. We worry about the long-term stability of the delivery system because, of course, it's essentially a surfactant. So is this going to, is this going to emulsify, for example, when as it's degrading? And then how does it degrade? And I think the biggest question for us is, can it be used to deliver a protein? And that one certainly isn't completely clear right now. So we were also at the same time working on another series of in situ gelling hydrogels. So these ones were thermally based. So rather than being based on contact with an aqueous system, these ones were based on the temperature change between the air and the inside of the eye. So they needed to gel somewhere between 20 degrees Celsius and 37 degrees Celsius. And we needed, we had this whole list of things. We needed it to be transparent. We needed it to be resorbable. And of course, we needed it to deliver the protein that we wanted over a period of 3 months at the very least, but hopefully 6 months or even longer, and then to degrade after that. So we started out with just a slightly modified version of the thermally gelling polymer polyanisopropyl acrylamide. And we incorporated all kinds of functionalities. So it was a grad student and a postdoc who worked together. So collaboration was really key in this project to incorporate things like, could we bind the drug to allow for the release? Could we incorporate degradability so that it would be cleared from the eye? And could we make it synthetically flexible so that we could manufacture it? So a little bit of science here. We took the NIPAM. We incorporated a functional group, an NAS group, that allowed us to bind things to it. We incorporated an acrylic acid group because that way we could control what temperature it gelled at. And then this DBA group essentially degrades. So we have a ring opening that occurs with hydrolysis at 37 degrees Celsius to create polymer chains. And those polymer chains, if they're an appropriate molecular weight, will just be cleared from the eye. So we get LCSTs in this case. We can play with this, but the initial materials had LCSTs that were maybe a little bit low, but molecular weights were in the right range. We were aiming for kind of 30 to 40,000. So, with some synthetic techniques, we were able to make polymers that showed some promise. And we, again, placed these into various systems. So we looked at degradation. And these things degrade. They're incredibly stable, certainly in just an aqueous environment. We only lose about 10% over about 3 months. And same thing with mass loss. When we do dexamethasone release, again, we find a burst, but a relatively long and stable period of release. But the release is a little bit high, but this burst might be okay. So again, potentially interesting from that perspective, and histologically very quiet, nothing that would show anything that would make us say we can't put this into an eye. These polymers, however, weren't clear, so we wanted to incorporate transparency in them. And again, this postdoc and this PhD student creatively came up with a bunch of ways that they tried out, and you can see here it's a lot of iteration. I'm showing this slide for this reason. But they incorporated polyethylene glycol into this system and made a whole bunch of materials with a whole bunch of different compositions. And by and large, most of them didn't work, but some of them did. So we did end up with a couple of gels that were stiff and transparent. So we moved forward with these systems. And like I said, we have optically transparent, and as a bonus, these gels were non-shrinking. So when you think about when this polymer system would gel, typically it would squeeze in like a sponge, but these systems actually held their volume, which meant that sponge effect wasn't going to occur, and we weren't going to get that big burst of drug. So same thing, we did some degradation studies, some enhanced degradation studies to show what happens when they degrade. And I think this was one of the most interesting results I've ever had in my career. We we put this system into the back of the eye, and did some histological studies. Obviously, did a sham and then an unmodified control. And the histologist that looked at these pictures said that he couldn't tell the difference between our NIPAM materials that were injected into the eye and the untreated eye. He could tell with the injected eye, but he couldn't tell with our poly-NIPAM eye. So that certainly, gave us hope that maybe these things had some potential. We could play with things to control the polymer properties, control the composition, for example, to get, different amounts of water in them, different gelation efficiencies, different degradation kinetics. So lots of good things as a result of these materials. Like I said, we could control the degradation kinetics. We could control how long it took for these polymers to lose mass. So if we wanted them to be degraded in, a month or 3 months or 6 months, that certainly seemed to be possible. And this time we had the papers, but we also had the patents in place. So all of everything seemed to be in place. And then we spent the money and we bought some protein. And it turns out that we don't get release kinetics that really will allow us to postulate that this might be useful for a 6-month period. So this is dexamethasone. Small molecule, hydrophobic molecule, should release relatively slowly. And in fact, this nice linear release curve would suggest that it is a hydrophobic molecule, but we're only getting releases of 2 to 3 months with this small molecule. We spent the money and bought the protein, and we saw a huge burst, so probably protein that was in that outer layer. And then we see a nice linear release, but this linear release, we've got 35% of that protein released at 15 days. So what that tells us is we're probably not going to get out to 6 months, 180 days. So, we've been looking at ways of incorporating other functional groups, but sometimes scientifically it just doesn't work out, or it doesn't give you the properties that you want. So you do a whole lot of work, and I mean, great papers can come out of it, but realistically, when you look at the actual application, it doesn't seem to bear what your initial studies showed. So I'm going to tell you now about a little bit more positive story. This is the third technology. And again, we've chosen the technology based on the economics and also the patient impact. In this case, we were looking at the front of the eye because the front of the eye is much easier. Everyone puts drops in when something happens with their eye. In fact, 90% of all ophthalmic conditions are treated with eye drops, but these drops simply don't work very well. I'm sure that, if I could see you all and I asked you to raise your hands, I'm sure that everyone would raise their hand when I asked you how much you like putting drops in your eye. Of course, I just love it. You hate it, right? I mean, everyone hates putting drops in their eye. Children are the worst. I don't know if you have kids, but putting drops in a child's eye is just about the worst experience ever. But even that, even aside from not liking doing it, just the way the eye is designed, you lose 90% of that drop almost immediately. So you blink, you lose 95% of that drop. So only 5% of the medication that you're paying for is actually getting into the tear film. And anything that's left in there can be exposed to the systemic circulation, can be absorbed, carried away. So you're losing even more in that case. A lot of these drops have to be given once a day, twice a day, multiple times a day. And that can become uncomfortable because a lot of the drops have a variety of different additives and preservatives that are good for the drug, but probably not good for the ocular tissue. And all of this leads to really poor patient compliance. So, you're losing 95% of the drops that go in, and you're losing 100% of the drops that actually don't get into the eye. So we were looking at what conditions we could treat, and we explored a whole lot of conditions. And this was around the technology that we had, but the one that we looked really closely at in 2015, 2016 was the dry eye market. Again, it's about the numbers. It's a growing market. A lot of people are presenting with dry eye. In fact, it's the most common reason for people to go to their optometrist or their ophthalmologist. And I would be surprised if these numbers aren't even higher as a result of the pandemic, because of course, we're all looking at screens all day, every day. So we have a mucoadhesive micelle technology. We were very careful to make sure that the patents were in place. And we initially saw this as a platform with many potential applications. But like I said, we chose dry eye disease as our lead indication, delivering the drug cyclosporin A, which is currently available in drop form as Restasis from Allergan. So the idea behind this drop is that we go from delivering these drops once a day, twice a day, to delivering them once or twice a week with enhanced efficacy. So there's more drug there constantly all the time. And the ocular irritation is much less. The formulation for cyclosporin A is actually very painful. And the other thing that patients who have dry eye disease who are on cyclosporin A report that it takes 3 to 6 months for the drops to actually have any effect. And we thought that by delivering a higher dose, we could potentially improve this period of time. So we could see a result in maybe a month or 2 months. So as we did with the other systems, we did, early safety and tolerability tests, in this case in rats and rabbits, and we saw that there wasn't really any signs that the eyes had any issues with the micelles. We've since done even more studies, and actually those have borne out, from what I've heard. They're not complete yet, but they've borne out these initial studies. So of course you want to compare whether this system is clinically efficacious, whether it compares to the clinical standard. So what we did was we induced dry eye disease in a rat model, put these drops on the eyes, either Restasis twice per day, which is the clinical standard, or every 3 days. And then we incorporated cyclosporin at a slightly higher level into our micelle formulation, and delivered it with the same frequency as the Restasis drops, so twice per day, delivered it every 3 days. And then we're wondering if we could go out to a week, but we delivered them every 5 days just to see what would happen. So again, early results, we learned a lot from doing these studies, but the studies certainly showed that we might have something here. Restasis twice a day, not surprising, resulted in a return of the tear volume to baseline levels. And the same thing when we delivered the cyclosporin A from the micelles twice a day, so not unexpectedly. However, when we took the Restasis out to delivering it every 3 days, significantly less efficacy, and in fact, very similar to the untreated controls. Whereas if we delivered cyclosporin A from our mucoadhesive micelle formulation, so a formulation that bound to the front of the cornea, we saw efficacy by delivering once every 3 days. And even though once every 5 days didn't show return to baseline, certainly there was evidence that we might be able to increase the duration between drops with these patients. Not a clinically indicated metric, but we also looked at corneal staining. So one of the things that happens in these patients is their corneas, they lose that outer epithelial layer. And again, very similar to the previous results, Restasis every 2 days showed a return to baseline staining. But in this case, all of the micelle formulations administered twice a day, every 3 days, and every 5 days showed a return to baseline in terms of staining. We wanted to look at, so we learned one of the things that we really needed to do, and one of the things we've learned is that no matter what data you present to people, they always want more. But we found that one of the key studies was that we needed to do a pharmacokinetic study. So figuring out what concentrations of drugs were in the tissues. So we delivered Restasis, again, twice per day. And then we put our micelle formulation on twice per day and once every 3 days. Slightly higher concentration this time, because as we've played with it, we've learned how to increase the concentration of the drug in the micelle formulation. And I think this is a really striking result, at least for me. What we found is that when we compared the concentrations of drug in the cornea, we found that compared to the Restasis amount, when we delivered the micelles at this higher concentration, we had a much higher amount of the drug in the corneal tissue, which would indicate that perhaps we could see efficacy in a shorter period of time. Obviously, that would come to bear clinically. And when we took the duration between doses out to every 3 days, the corneal bioavailability was very similar to Restasis twice per day. So it does seem to suggest that these micelles are binding to the eye, they're adhering, they're providing sustained release, and this is leading to a greater amount of drug in the cornea. So all of this seems really positive. So we've taken this, we've spun out a company, as I said. We've done lots of work, and when I say we, I'm saying that rather generously, because as an academic, I think one of the key things is having a really strong team around this, a really committed team. And, you know, kudos to the team. And in particular, in this case, it's been Francis Lazowski who's done much of the legwork around trying to figure out what we need to do and where we need to go. So, you know, we've developed this whole strategy around can we deliver this in, you know, with cyclosporin to treat a variety of conditions, including dry eye disease. We've also been looking at partnerships with companies, for example, who have a small molecule that wasn't quite efficacious, or that they have trouble solubilizing, because this system will solubilize small molecules. And we've been doing some work for some companies looking at different molecules and their incorporation into the drug delivery system. Like I said, lessons learned. The team is absolutely critical. I think the senior academics amongst us would recognize that we don't really have time to put into making a company really successful. You need a committed group of people around you to help with this. And I've been incredibly lucky. Like I said, Fran has done the bulk of the work. She speaks business now much better than I ever could hope to. And then kind of on the scientific side, we've got a really talented chemist who looked at things like CMC and that kind of thing. So did some early proof of concept studies with manufacturing. And then Ben, who's done most of our in vivo testing. And the other thing that I would say is absolutely key to moving things forward, and something that you want to get in place early, is a group of advisors who will tell you up front whether because you start to believe your own stories, you start to believe your own BS, as we say. And these people, need to be able to say to you, look, like, that doesn't really make sense. The results don't necessarily follow what you're trying to say. What if this, what if that? And we've amassed this incredible group of advisors over the period of 4 to 5 years. And they've been, I think, absolutely key to helping us understand what we need to do, helping us understand what we haven't done yet. So where are we now? We've done all of this with non-dilutive funding. Much of the Ontario Research Fund grant, the C2020 grant, was really focused on moving this technology forward. This was the one that we thought had the most legs. And so we spent a lot of the money on that. And then we've generated some funding from the NSERC Eye-to-Eye program for the chemistry. We had some funding from the MSc POP program from the OCE that allowed us to work with Talina and allowed her to do some of the chemistry. So where we are now is we really need external funding. We've kind of exhausted what we can do with the non-dilutive funding from the government. We've exhausted what we can do within the academic environment. So that's something that you want to think about, is that is going to happen. You're going to need investment. So you want to make your decisions very easily or very early on what kind of data you're going to generate to get what you need in order to move things forward. So there's still more questions than answers, still very scary. I think we're busier than ever with this. So what disease do we use? Is dry eye disease still the place to go? What are the endpoints? That's really critical. What is the clinical relevance? So we have to have both the economics and the clinical relevance in place. Academic data, we've got great academic data. I could publish some wonderful papers out of this. But when a company digs down, do we have enough to pitch to a commercial company? Manufacturing, how do we manufacture these things? What's the benefit of keeping it close to your chest versus getting it out there? And how do you patent in that environment? So, we've had people say to us, well, people would be knocking on your door if it was a wonderful thing, but we've held it pretty close to our chest because we've been very cognizant of the patent issue. And obviously you want multiple patents. So we have kind of the base patent, but it would be nice if we could kind of picket fence around that. And then the big question is, how do we fund this? So all of the next steps are going to require funding from someone external, and how do you actually get that funding? And those are questions that we're grappling with right now. So in conclusion, it's a long road. It's longer than I would have thought. When I had those 32 technologies at the end of the NSERC 2020 network, I thought, 5 years, right? Some preliminary clinical, we wouldn't necessarily be in the clinic, but we would certainly be in clinical trials by now, which is 6 years later. We are not. We would love to get some first in human data, but we're not there yet. I would say the team is absolutely critical, and in particular, young scientists with a thirst for and a passion for entrepreneurship is really key. I've also learned that business is very different from science. I can sell something from a scientific perspective, not so good at selling it from a business perspective. And then, kind of what's next, and some of that what next is very scary. Actually putting this in an eye is very exciting, but it's also very scary. So I'll end there. I just want to acknowledge the incredible team of people, and this is just a part of them, but these are people who have worked on these 3 technologies over the years. And then I showed up our advisors, but really our key advisors have been Lynn, Lyndon, Michael, and David Wong. And then our funding sources. So we've been pretty lucky to have some good non-dilutive funding, but being able to now capitalize that and change that into an actual clinical product, that's the big question. So I'll end there, and I'm happy to answer some questions. If I can figure out how to stop sharing my screen.
Thank you so much. Yes, thank you so much, Professor Sheardown, for a fantastic talk and inspiring journey in your academic and also entrepreneurial career. So we got some several questions in the Q&A box. Before Mohsen asks his question, I will go through the audience questions here. Amir is wondering about how you administer these kind of hydrogel in the back of the eye. Is it
they're injectable.
Syringe
they're
or
injectable with we actually made that one of the criteria. They had to be injectable with a 30 to 32 gauge needle. So
Okay.
that was one of the criteria. And clinicians routinely use up to a 27 gauge needle, so we're fine. We've got good injectability.
He's also wondering, how you make sure. I mean, how you measure the degradation. Do you sacrifice the animals?
We didn't actually do in vivo degradation studies. We did those based on the ISO and ASTM standards. and did accelerated degradation studies in vitro.
Okay. Nike is asking, what kind of instrument did you use to determine the concentration of drug that was released from the
I've got an amazing research engineer who has lots of experience with drug delivery systems. So she's done all of our loading and release studies, measuring. She measures how much goes in based on obviously mass balance, but she also does release studies to determine. So a combination of both.
Thank you so much. Amir is asking about the toxicity of this material or cytotoxicity. Did you check it?
We did in
vivo
vitro in all cases, but I really quickly moved from doing in vivo tox with the things that we've done. So we've looked at toxicity in vivo. Early on, we were doing mice and rats, but we've moved to doing our toxicity studies in rabbits based on the advice of our clinical people.
Thank you so much. The other question is about what are the challenges of designing the biomaterials for drug delivery into the eye using the LCST polymers. The transparency is important, mechanical property, or what?
Yeah, so we first of all said it had to be a polymer that was acceptable in the ocular space, and then immediately broke our rule using polynipan because it at that point wasn't accepted in the ocular space. It has since been used in a couple of delivery systems, so it's becoming more acceptable. I'm not sure if there's FDA approval for it yet, but a lot of it was trial and error based on an understanding of the properties of the polymers. But yeah, like degradability, searching through the Sigma catalog happened to try and figure out what might work, what might polymerize with the system that we had. And I had a pretty talented postdoc who was a really good chemist, and that certainly helped a lot.
Great. Ali Reza is asking about this LSCST of these polymers you show in the table, and he's wondering if you get any needle clogging during the injection because the LSCST is about 20 or so, and for these hydrogels.
Yeah, not a huge issue. Like I say, we designed them so that they would be injectable at 30 or at 20 to 25 degrees Celsius, and not a lot of needle clogging. Obviously, if they sit for a period of time in the eye, you will get clogging of the needle. But if you load it up, put it in, and inject it right away, it's pretty seamless. And I think that's a practice thing. I think Ben has gotten very good at doing that work.
Great. And the last question in the Q&A box, I also encourage everybody to put their questions in the box if they have. Amit is asking about the drugs that are being used in this field for the eye. What are those drugs that we can deliver to the back or in front of the eye?
So yeah, front of the eye, we've done a lot of work with steroids, a little bit of work with antibiotics. Basically, our criteria, because we're an academic lab, is that it has to be cheap and off patent. So we've picked drugs that are available through Sigma or through other chemical companies and that are off patent, because we don't want to be stepping on any drugs. We've looked at dry eye drugs. So all things that, are used clinically. I mean, the spectrum, to be honest, is not that wide. I mean, there aren't lots of things that are used. So it's pretty easy to pick something that will work.
Thank you so much. Now it's the time for Mohsen's question.
Well, thank you very much. So before I ask my questions, so Heather, I think we can see your screen. Your screen is shared.
Yeah, I can't figure out how to turn it off.
So there is Oh, where is it? There you go. Oh, we lost Heather.
Are you with us,
Well,
Heather?
I mean, no, I think the actually the battery was also running low from what I
So can
observed.
you
So there's a good chance
invite her
that.
again, maybe? Ah, Heather is here.
Okay, so Heather,
Yeah.
let me
Could you
invite you
invite
again
him?
then.
Yeah.
But I noticed that the battery was running low as well.
Yeah, I fixed that.
Okay.
Can you see me now?
Yeah, I was like there, like very nervous. I said, okay,
I
okay.
couldn't figure out how to stop sharing without losing the
No, the
video
battery.
off.
I was nervous about the battery. It was the number was going down. It was like 14 when you started, and it went to 6. And I said, oh, it's going to stop working. But well, thank you. So Heather, amazing talk, very translational, and then we do some work in the area of translational research in wound management, and I kind of relate with what you mentioned in terms of like patenting. I've made those mistakes. Chris Flores is, I think, our industrial liaison is on this talk, and probably he knows the mistakes that I've made, and then other aspects, especially the in vivo studies and in moving things forward to clinical studies, which we are also very scared. Just to confess, it's not something that we are used to. I mean, clinicians are very easy. I mean, they know, they're familiar with the process. I'm not, so it's a scary, field for me as well. So I can completely relate with that. So if you don't mind, I have a few questions, a few technical questions, and then a couple of questions about the translational aspects of your technology. But I'll start with the latter. So you mentioned at the beginning that you look at the market. So there's a big market there. It's a $40 billion market. The majority of the market is, I remember, $23 billion is anti-VEGF therapies. So there's a big market there. But are they big players in the market as well? And then if there are big players, what are your commercialization strategies when you want to deal with like a giant Johnson and Johnson, company that is, that can, get the technology, and then competing with them is not going to be very easy.
Yeah, there are definitely big players, and I mean, we talk to them, of course, but we've also done well talking to some of the European players. So just talking to everybody. I mean, in the ophthalmology field, there've been a couple of new systems that have come out from smaller companies, and they very often get swallowed up by the bigger companies, but they haven't yet. They're still kind of operating on their own. So I see that as being what might happen, is that a smaller company might take it up and move forward with it, and then they would ultimately be absorbed by somebody a little bit larger, because the big players are obviously huge, and they they have a lot of reach and a lot of scope.
Okay, that's fair. So when do you plan to engage them? At what the stage of your product development are you planning to or you envision to engage them into into the conversation that they probably license your technology and then just distribute it?
Yeah, so we've been engaging them right from the start.
Yeah.
Probably a little too early, but it's it's always good to talk to them. It's good to get their feedback. Sometimes that's a little depressing because they have only negative things to say, but they're also brutally honest, which is really nice. So I mean, we've really engaged with people right from the start, and I think that that's a good way to go forward. There's, in the ophthalmology field, we're pretty lucky. There's a couple of fairly major meetings that are commercially oriented So it really is an opportunity to talk to everybody who's playing in the space.
Mhm. Okay, that's fair. I mean, we had the same situation before, but so now my question is, when do you, so at what stage do you think they will be interested in, in talking to you about licensing the technology? After the first phases of clinical trial, or after?
Probably, yeah. We have to have some clinical data. That's what we've learned, is that clinical data are key. So we thought that, we thought that we could get them earlier, but realistically, we need to have our first in human data, at least to show safety in a human.
Safety.
Yeah.
So speaking of human data, I think you need to go through like approvals from Health Canada. This is, I think, or FDA, before you can start your clinical studies or in human studies. So my question here is that in terms of safety, what considerations do you need to have before you get into get approval from Health Canada, and then also what class of medical device is your technology is being considered from the Health Canada or FDA point of view?
Yeah. So it's not a device, right? It would be a drug. So slightly different from that perspective. We look at this as a formulation, and it's just a matter of ensuring that the formulation is safe. And getting those first safety data are actually quite difficult, because we can't just put the formulation in. We have to pair it with a drug. And the problem with the dry eye drug that we're looking at is that it can take some time to show efficacy. So, these might potentially be long studies. So we've looked at other drugs that might have a shorter period. Where, we could measure something almost immediately, right? You can measure the intraocular pressure, or you could measure something almost immediately. And I think that's really going to be key to us getting into the eye. And then, like looking at partnering with other countries, for example, that have better more lenient or more open policies around how do you put something into an eye to get those initial data.
Okay, it's very interesting, because I always wondered these, especially the gel-based technologies, if they are considered as implantable devices or drugs. So in this application,
the implantable devices may be considered a device or a combination therapy. So we'd have to show, which is why we really have moved forward with the micelle formulation, because it really is a formulation.
I see. Okay,
so
that's fair. It's very interesting. So this is another aspect that when we want to come up with these technologies, it's good to know how you can move things around to in terms of obtaining the approvals from the regulatory, from for example from FDA or Health Canada or European Union. This is very interesting, and very for me, it's at least I'm learning this, because this could be very it changes the direction, the path that you move forward. So you talked about multiple patents, which I recently learned is good if you want to have a company, and then if you want to attract investors, they like to see multiple patents or a patent portfolio in the company. This is something that we've been discussing with our team. But the issue with having multiple patents, in my opinion, is the cost. So how to keep them alive is pretty costly. So if you have like a company with 5 different products or 5 different patents, then you need to keep them alive, and you have to pay for them. So what are your thoughts on this? You briefly mentioned.
Yeah, we've grappled with the same thing, because we can't obviously afford to pay for multiple patents. So something that we've looked at, and I mean, we have a McMaster owned IP policy at Mac, but my research is not going to pay for multiple patents. So Obviously, we need investors in order to make
Indeed,
that
indeed.
happen.
This is something to PI to PI.
Yeah, for sure.
It is very difficult. And sometimes you need to sacrifice some of this patent at some point, right? There's like you have 5
absolutely.
patents. And so how do you decide on, and again, the reason I'm asking this is because we had this issue and then we had to at some point drop some of the patents. Patents. So how do you decide which patents, which are our kids, or I don't know, these are the technologies that we to drop
I'm
them?
hoping that I don't have to decide. I'm hoping that the licensee will make that decision for me.
Yeah.
I mean, right? Like right now, we have potentially a second patent that we could submit, but we're holding off just because we don't feel like it's the value isn't there, right? I mean, the cost to benefit ratio is
That's another complication. Yeah, I mean, so you have this patent now ready, and then there probably a trainee who also was involved in developing this patent. Probably, I don't know, but in our case, so they want to publish the data. You have this patent that you are holding up. You're waiting for the right moment to file it. So how do you balance all of these, or what are your thoughts on these kinds of,
Yeah, that's tough for sure. I mean, we've done, we've held back stuff for sure. With the micelle technology, we haven't done any publication since the initial 2016 publication. I think there's at least 3, possibly 4 more publications in the data that we have. And it's just a matter of, when we release those publications. So we haven't had trainees working on this. Fortunately, we've had some postdocs, but they understand the entrepreneurial space and they are okay with that. So I've been pretty careful about hiring people who understand that, you might not you'll get papers eventually, but you won't get them right away. But yeah, it's a tough thing to do. So I try and keep my early stuff with my grad students, and then when we're doing the much more translational stuff, it's with postdocs and research associates and research engineers.
Okay, that's fair. So Heather, I still have a few questions to ask, but I wonder if you still have time.
I'm actually a little short on time, but I can take a couple more.
Okay, all right. So are we good with like another 15 minutes or
Yep.
I can stop anytime that okay, that's great. So there are new technologies being developed in research labs mostly, which are incorporating sensors, for example, in lenses, smart lenses, and these kinds of technologies. What are your thoughts on those, technologies?
The idea is attractive from an academic perspective. Again, it's about, like the 2 things I said, like the economics, and then also the patient, right? The patient need and then clinical efficacy. So, for example, Google spent a whole lot of money developing a lens with Alcon that sensed glucose in the eye. And it turns out that ocular glucose concentrations do not necessarily mimic those in the blood. So it might have sensed glucose perfectly, but it really didn't give a clinically relevant number. So, I think that that's really key. And I'm not sure that you need to find the right indication. So, I mean, pressure has been suggested as a potential indication. I don't know. I can't off the top of my head think of an indication that is perfect. So, I mean, that's really the key thing. I mean, when a company as big as Google partners with a company as big as Alcon and they fail as epically as they did, obviously they're making mistakes too.
That's probably is something that it's probably good for us at least. I mean, as if we make mistakes, I mean, big companies are also making mistakes, which is okay. I think it's a
And our mistakes are usually publishable, right?
so. So I have a question about the NIPAM-based polymers that you're using. So you mentioned that there's no issues with clogging with the needles sizes that you're using. So but in terms of the burst release, I wonder if that is because of the loading of the drug during or that's the another issue. So for that reason, my question is, at what stage during the fabrication process do you load the drug? Is it after you create the polymer and then load it with the drug, or it's during the synthesis that?
No, it's so we synthesize the polymer,
Okay.
and then we mix the polymer in an aqueous solution with the drug.
With the drug at low temperature, and then okay.
Yeah. Yeah.
Okay. So could it be possible that the drug on the surface, which is closer to the surface of the gel, releases first, and then starts?
Oh, absolutely. That's what's happening. Yeah.
Okay. So that's probably okay. All right. So my last question is about the advisors. So you had a list of very stellar advisors that you that I think is essential for any company, a startup. But at what point do you bring them on board? Is it like at the ideation stage, or is it like at the later stages when you have done some some preclinical studies and you want to just discuss them with them?
So I think it's a combination of both. I've been pretty lucky in that and ophthalmology is also a very academic company partnered kind of area. So at our annual meeting, there's always lots of companies there, but I've been pretty lucky to have met company people early. The 2020 network basically forced me to bring in some company people. So to kind of with them on things that were really early. I've found that company people are pretty interested in some of that science stuff because they don't get to do it. There's a lot of PhDs still in companies that don't necessarily get to play in this space very often. So they like that scientific side of things, and as we all know, people like to give advice. So that's also played in our hands a little bit. One of the advisors, we actually hired him when we started the Ontario Research Fund grant, just to bring in an outside perspective, another case of believing our own stuff, because it's really easy to convince yourself that what you've developed is the next best thing since sliced bread. And it's really nice to have that outside perspective and go, well, there's some issues here, and this is something you should maybe think of. Yeah. So, and then he's been great about introducing us to people. I mean, we've just, created this network, this web of people. So I would suggest talking to people early and getting your name out there, getting your face out there, making it so that people know who you are, and building on that, because like I say, people like to give advice. So. Okay,
that's very good. That's good to hear. That's good to hear. So I said that was my last question, but I have another question which just came to my mind. And again, I'm very interested in this topic. So my question is about the age-related macular degradation, which you showed that this could be one of the applications of your gel that you inject in the back of the eye. So how often do the patients need to inject drugs right now? I mean,
So when the first drugs came out, they were injecting them on a monthly basis. And then patients started, or our doctors started with some of the patients going to a treat and extend regime. So basically, they would see if they could extend them out to 6 weeks and then 8 weeks, and then some of them out to even 3 months. There's a subset of patients for whom this works, but there's a subset of patients who really do need those monthly injections to keep the disease stable. There's a couple of new drugs coming out. So one that's been out for a while, and it works best on this treat and extend regime. The original drugs do not, they much more require that regular monthly injection. There's a new drug coming out within the next year, I believe. So clinical trials ongoing, good results, and they suggest that it can be every 3 months. But the doctors, from what I understand, are a bit skeptical about that. They still want to make sure that the disease is managed, because if the disease is poorly managed, things can go south really quickly. And once the vision is lost, it can't be regained. Yeah. So they want to maintain, concentrations in the back of the eye that are therapeutically efficacious.
And this is these injections are it's a lifetime injection, so they have to do it every.
Yes.
Okay. So in terms
which makes it a problem for other diseases like diabetic retinopathy. The symptoms are the same, the drugs would be the same, but the problem is when you've got a 35-year-old diabetic patient, you don't really want to be looking at, another even 50 years or 40 years of injections once a month. That's the incidence of complications is not high, but it's high enough that this would be a problem.
I see. So in terms of these gels that are injectable, so do you want these gels to be degraded? Like, let's say if the goal is to achieve sustained release for, let's say, for 6 months, do you want the gel itself, the vehicle, the drug vehicle, the NIPAM drug delivery system, to be degraded over that period as well? Because in the next injection, you want to inject another gel into the eye, and if they don't degrade, you will have accumulation of drug, gel. So that's the idea here.
Well, I mean, the idea is that it releases stably for that 6 months, and then the degradation by and large happens after that. So there are systems that are non-degradable that are placed into the eye, and they're placed once every couple of years. They never go away. So there is a history of things accumulating in the eye. I'm not sure what the long-term impact of that would be, but I think something that degrades over a year, for example, would be perfectly acceptable.
Perfectly acceptable. So there's no need for like a secondary surgery to remove them.
No, and that's what we were aiming for, was not having to go in and remove them.
Okay, that's fair. Very interesting. So anyways, I mean, I was very, very excited about this talk. It was very informative. Thank you very much. I don't have any other questions to ask, and I know you are very busy and that you have to leave us quickly. But thank you very much for your very nice presentation. I really enjoyed it.
Well, thank you. It's been an absolute pleasure.
Thank you so much, Heather. And please don't forget the audience. Next Wednesday, we have Tamer Mohammed from Aspect Biosystem. He's talking about 3D bioprinted cell therapy. So see you all next week, Wednesday at noon Eastern time. Bye-bye. Take care.
Bye, everyone.